take that sequence and put it in E. coli, it's likely going to work. It's at least going to read for the protein that we're after. So we'll have to do a little bit of RNA extraction, freshly, just RNA extraction right there. Then do reverse transcriptase PCR, right? Extract it out, make some DNA copies, probably send it off for sequencing to make sure. Yeah, get that sequence, and then we're going to be plopping that into our vector. I saw a hand. You might want to ask another question. Yeah. You can choose. If you would want. You can actually get it right. All right. So one of the things that I like to get in my plasmids is having a differential marker, and I'm marking this one blue because it's for the LAC-Z gene, and that's for beta-galactosidase. Lots of big words, but basically, if this plasmid was put on a plate with a special agent called X-gal, the E. coli that had this plasmid would turn blue. Okay, the E. coli that didn't have this plasmid would stay white. That's really very common. Okay, I'm going to be using X, gal, it's white, blue selection. All right, so one of the ways I could find out that at least my gene got into the E. coli and was making more copies of it is probably just do a little switcheroonie between my gene of interest and the LAC-Z gene. Does it have to be like right on top of this gene? Does anybody remember from last class? Does it have to be like, does it have to switch from like right here? This one gets cut right here. Does it have to switch out perfectly? No, you just have to interrupt this gene. So put it in somewhere, anywhere in the code here, we'll make it so it doesn't work. Preferably in the very beginning part, right, promoter or otherwise. It just needs to be interrupted. To do this process, we would take a restrictive enzyme, one specific one. So A, restrictive enzyme, B, right? And we make sure it's the same orientation as this one. So let's pretend like this is the arrow. So beginning, right? This is the beginning of the arrow. Restriction enzyme B, restriction enzyme A. Bada boom, bada bang. It will get in here. Now, something that can happen with this process is that when we cut our vector, not all the original pieces that get cut. Not 100%. It doesn't happen. Not 100%. It doesn't happen. And the same thing will happen with our gene of interest here. Okay. So in our reaction mix here, we're going to have some whole pieces. Okay. I'm going to color code this in a second, folks. And okay, we're going to have completely uncut. Ooh. Hopefully people aren't super colorblind. If anybody's kind of colorblind here, just ask a question so you just can't see it clearly and I'll explain what you're seeing. I realize some of these colors aren't good for everybody. That's what I have. Okay. Okay. All right, so what you're seeing here is you're seeing plasmid uncut. This is our gene of interest. I'm sorry, I switched sides. I didn't realize actually it's not helpful. There, gene of interest. Flat Z. So I cut out my plasma and all of it's cut, so I have these two parts in the same mix. I have some of this that doesn't get cut, but you can't really tell much of the difference. In the end, I want this product here. So what you typically do is you have your cut plasmid, and you're going to mix it with your gene of interest. These sticky ends are going to attach to each other. You're going to add another enzyme to get your final product. Sometimes you can choose to purify your cut product on a gel electrophoresis, which separates things by size. So you're going to get two bands. You're going to get the part you want, and then the laxi, which is just going to be further down because smaller pieces migrate further. And you can cut out this part, right, from the gel and purify it out that way. This can maybe interfere with your later process, so sometimes you have to skip that step. And so that's one of the reasons why we knock out a gene, so that you can know that this piece got into your vector here. I know, super exciting. Okay. So we use a differential marker here to prove to ourselves that we want white colonies.
I was going to ask you a quick question. So when you go and knock out laxi, you say you can cut it anywhere. So basically, while it's reading the DNA, like you'll have the start codon, all the sequence, and then suddenly you'll just get another start codon for our gene of interest, and it knows then to not. It just
it'll make a product that doesn't make the beta-galactosidase anymore. If it doesn't make that, it could stay. It can be trash.
Okay.
Yeah. But it doesn't make a functional gene, and so we know that something is successfully interrupted. That's another great reason to use two different restriction enzymes. It's because these ends won't re-meet. They won't meet unless they're two different blunt cutters, and then you're not doing any specificity at all. It's like, two flat ends will always meet, no big deal. But the edges here are kind of like cut keys, and they only fit in, or like jigsaw blocks, right? They only fit with their correct partner. All right, cool, we got our gene in. But I don't know if my gene is being made or not, right? I don't have an idea if it's being made. And so one of the ways that we can kind of determine whether the gene should be being made is that we add a little tag on the end. And that tag is actually green fluorescent protein, or something else along those lines. Green fluorescent protein is something that you can see doesn't tend to affect the folding of many genes in practice. So we can put green fluorescent protein in here so we can know if it's being made. Just got to write it here. It's not really false negative, but if GFP is being made, then you know the message RNA is being made, and that essentially that your gene here is being translated. And the way that we know it's being translated is that you tend to put your GFP at the end of your gene. So the start ATG, methionine, is on this end. And at the very end, you have this tag. It tags along at the end of GFP.
And you don't need another promoter. You can just use
No,
that
you want to make sure that it works under this promoter.
Okay.
Now, an option here is that you want to also make sure GFP can be seen. You want to have that as a control as well, because it could just be, right, it could just be the promoter. Promoter you're selecting, or how close it is to the promoter, or something else that's going on, could be affecting that you're not getting GFP. So when you're not getting GFP, so this is actually false negatives. So no GFP. And you have growth, that could be the promoter. Or a translation issue. Or, yeah, GFP doesn't work in that cell for some reason. Or you're not exciting GFP at the right frequency. The right wavelength. So it's always good to test all of your parts before you move forward. So indeed, you might want to test this promoter and its efficiency by just having promoter GFP. And then in your original test, why not? You can switch out the GFP, but it's better to... It becomes complicated when you're using GFP as something to knock out and also to tag your gene. It'd be nice to make sure that GFP works and just works in your lab. But I wouldn't necessarily put my gene of interest with GFP as its tag into a vector that has GFP as a tag. It could get confusing. It could be something else that's going on, and you want it to be only one thing potentially going on. So to test the promoter, you can either have the same promoter behind your ampicillin resistance or another reporter gene. So maybe you can use red fluorescent protein or something like that. Thank you. To test to see that it's just the translation, that for some reason something about the ribosome binding site, something isn't working, or this gene is like toxic, or something else is going on, you'll need to see if the messenger RNA is being made. I would probably send it out because that's a pretty sensitive test, and it's highly subject to contamination. All right, so you are seeing GFP, but are you getting expression? There's two things you can do. You can test it out to see if the messenger RNA is being made for your gene of interest. So are we being made? Let's see here. Okay. So message RNA. Check that. You want to maybe also extract the protein, which is really hard. Something that we had one class in here, but there's other things you need to do to your gene to to ensure that it can be extracted. So you might even just come in here with, like, once you see GFP, you're going to assume that your gene is being made, and you may try adding the second gene to see if we get the lemon smell. imagine what else could happen in our experiment. So this is just an idea. Some of the things I would like to actually do a flow chart through this, so we could show all the steps for each step. So I want to erase the false positive and false negatives and then work through how we would do each part of that experiment. Is that okay? And I'm gonna post that picture so that you all have to see it. Because there could be other ways. Okay. So step one, we're going to take our cells, and we're going to see a grow on LD. As they grow on, as our example, ampicillin plus plates. Right? So if they don't grow on LD, we need new competent cells. If they grow on ampicillin dove plates, we need new competent cells. So grow. You continue, they grow, you don't continue, and you return to step one. All right, step two. We want to make sure our vector works, right? And so might as well just do our transformation with our competent cells. When we make our competent cells, we're going to have we're going to play 3 reactions. Okay? Can anybody guess what the 3 reactions are going to be? We want a false positive, false negative, and then one's going to be like
positive.
Our test, right? So one's going to be our test. And then we'll put false. FP for false positive, false negative. So what would happen in a false positive? So that means we think we got a positive transformation, but it wasn't a positive transformation.
The cells would grow.
Yeah. So this would be our competent cells minus the plasmid. I'll put parentheses around this so you know it's minus the plasmid, and this has ampicillin in it.
You mean the media has it?
Yeah, so you've got competent cells to grow.
So no plasmids, so no ampicillin resistance, right?
Right, but they grew anyway, okay? And then there's also another just positive control. And this is your just competent cells on LB, nothing else. False. Oh yeah, so this, and then you can actually, this is, if you want to be thorough, you're actually going to have 5 plates. And so this would be competent cells plus another plasma that you know works in your lab. Because we're actually testing this plasmid here that we've never used before. You can skip this one if you know your plasmid works. Common cells on LB. What is this one for? Okay. Let's make sure that our selective agent is working, right? Make sure the selective agent is working. This is our test to make sure our new vector works. These set of confidence cells went through the whole processes of the set of confidence cells that we know worked in the lab, plus a plasmid. I was down on the floor, I should have written it down. There's one thing that we didn't test for. Cells grew. Okay, it might just be 4 plates. Getting excited. Okay, so let's make sure our vector works. Because we want to make sure that your confidence cells didn't die in your experiment. All these cells get treated the exact same way. The only thing is what the cells are plated on, and also what plasmid the cells receive, if any. And so this is this is one. This is just a transformed E. coli that is AMP-resistant positive. Including that plate will ensure that you didn't put too much ampicillin, right? So this one here tests whether your transformation protocol didn't kill your experiment. This one here tests to make sure that the ampicillin was at the right levels so that an organism with the ampicillin resistance cassette could grow, right? Because that could be a possibility. If you're working in this lab, do all the controls. 4 reactions. I know it's 4 reactions. I know it's more to keep track of. I know. But it saves time. So when you get a result doing it this way, you know you can move forward. You've actually seen stuff. But it's nice to explain all the possibilities to yourself because you can move forward or be discouraged. And you don't know why. I think it could be you, right? So this guy's actually a false negative. Here, this is a false negative. This is a false negative. This is a false positive. You know something works. This is just a check. It's the rules against false negatives. So you can see that there's a lot of things working against you, obviously, because there's only one opportunity for false positive and a lot of opportunities for false negatives. All right, so when you do this next step, you do not have to prove to you can, but you can make plates all at the same time to help control for not having to do every single control every single time. We're going to assume that we have enough ampicillin and it's not too much. But for our next step, we will still need this plate. Because our differential agent make sure it doesn't kill our cells. And this also, you also want to make sure that if you have an inducer in the plate, that the inducer is working right. So this plate is going to be, again, pre-transformed E. coli, right? That is positive for ampicillin, positive for our case X-gal, as X-gal on the plate, right? And also work with that. And if there's an inducer to turn on the gene that we're looking for, there's inducer and reporter. So this is the one that has everything that we could possibly want as a feature expressed somewhere in our experiment. So that's our false negative, because if we don't see something, we need to make sure that it isn't something else going on. So something that works for all of the features that we have in our plasmid is important to include. We're doing the transformation, so we make sure our competent cells are working. You can do ampicillin here and also your inducer. And if there's a substrate, which this one happens to have a substrate as well, because you want these cells here to not grow and also not be blue. You can also put the inducer on this plate as well. If there happens to be one. You want to make sure it's not toxic to the cells. You also want to make sure that it doesn't cause your non-transformed cells to turn blue, which can totally happen, in theory. Our new plasmid is going to be differentiated from anything we don't want. So in this case, remember what we don't want is blue cells, because we'll have, draw our plasmid for us again. This other circle is a plasmid, it's not a petri dish. Even putting plasma underneath is not going to help us. Oh, we'll put little stripes in it. And our ori. Okay. So the plasma that we're changing, that we don't want, but there could possibly be in there.
Yeah, when are you coming back?
Has the ability to make blueness in our plates. That's why it's blue. Go figure. Easy. Okay. So we're going to be interrupting that gene and putting in our own gene. So the colonies that we want are going to be white, right? And blue colonies we don't want.
I haven't looked at anything else yet.
Is this making sense to people? I know it's a lot of just like little steps.
So when you're adding something to it that makes it turn blue, you don't want to have that grow the other.
So there's something in the media, right? It's a substrate for the LAC-Z enzyme. When LAC-Z works on that substrate, the colonies turn blue. When you interrupt that gene, the colonies don't make that enzyme anymore and can't turn blue. So you want to select colonies that are white. Yeah. So this is just an example. When you start doing your own experiments, you're going to take, like for instance, competent cells. You're like, I want to make sure, you will write this down, I want to make sure that my competent cells are ampicillin sensitive, right? But still can grow. And so you're right. So if this is true, when I plate my competent cells on ampicillin plus media, I will see no growth. Hypothesis 1, right? Hypothesis 2, if I plate my cells on just LB, they will grow because my cells are viable. Hypothesis 2. You can't know those things until you try it. You write it out. Second part of your experiment. I want to know, are the cells growing because they got my plasmid? So let's ask that question.
The plasmid contains resistant things.
Yeah, are cells growing
I think the hard part is, are we going to set some kind of form?
because they got my plasmid? The second question you want to have answered is, are there no cells growing because one, the protocol, the transformation killed him.
Since we're a project, we could do something
Killed.
creative, like,
Two, the antibiotic resistance.
present plus number of days of this.
Antibiotic, too strong. Three, DNA killed. So this is kind of in that one. Transformation killed. That's too many. What else could have killed my cells? Environmental conditions.
Contamination.
Let's say 4. The AMP. Yeah, that's good. AMP-R gene. No work. 5. Contamination. So it's kind of hard with contamination not growing. If we're like, well, it could have been contamination. If it's contamination, hopefully we'll see other things grow. But it could be viral, bacteriophage contamination, and they just kill the cells. You just don't know. But you still, it's like, are the cells growing because they got my plasmid? Is this first one. Now that's just like, cool. We can also itemize that one out as well. Because in order to grow, they should be AMP resistant and have the plasmid. These could be one and the same, but I'm breaking apart. Because the cells could be growing just because they're AMP resistant naturally, or they could be growing because they have my plasmid. Those are the 2 reasons why I think that the cells could be growing. Oh, or 3. My ampicillin is no good. Okay. So these are the 2 questions that I'm asking. These are all the hypotheses of what could be happening, and if the cells don't grow, something went wrong, right? I didn't have any success. These are all the reasons for a false negative, right? So when I'm troubleshooting, I'm like, all right, here's the reasons. Now, the transformation killed them. Well, that's why I do a control transformation. That's something that I know that works. Okay, do a control transformation. And then also, part of the transformation is making sure the cells are viable, so I make sure that I played competent cells that went through this process. I just played competent cells on LB just to double check. Because if this control transformation doesn't work and I don't have the competent cells tested, it takes another day, right? So you just do it at the same time, just to double check. I know it seems redundant, but redundancy is good. So that's the transformation killed it. That's a possibility. Antibiotic too strong, right? So we have a control transformation can answer that question. But testing for two things, so just keep that in mind. Or you could have already transformed cells. That are resistant. There could be environmental conditions outside your experiment that make it work. So you incubate your plate at the wrong temperature. Your E. coli do not grow at 42°C. They grow at 37°C, right? Or they do not get enough time to grow. You're not seeing them grow because you only waited 3 hours, and it takes 24 to 36 hours. Okay? So you just review your stuff. So there's a lot of reasons in the environment that you can try to control for, that can explain later, but you can't make an experiment for, or you could, but you'd be making tons of plates and having a lot of incubators, so you just don't do it. So the AM gene's not working. So that kind of comes back to the construct. So if, you know, all your other controls are working, right? So ampicillin resistance gene from that similar plasma, or that same gene is working in the plasma, you're going to assume it's working, especially, and it's like the ampicillin's at the right level, because your control transformation / your pre-transformed cells are growing, right? So you kind of, it's a tree, it's a diagram, you just walk through it. So this is all the thoughts I'm going to have before I even do the experiment, right? Like, what's going on? What could happen? So this question is actually answered by these 4 experiments here, right? Some of these overlap. So here's AMPAR's not working. Then none of the cells are going to grow. But you want to have something in the cell or borrow a pre-transformed piece, right? So basically you need those plates. But you understand we're answering just 2 questions here. What I hope to happen, what I don't hope to happen, and then explanations for why I'm seeing this and why I'm seeing this. And then a physical experiment that says what I'm seeing. Does that make it a little bit more clear? Okay.
So step 5, you go into the clean room and do a control dose.
Yeah, you just try to do better sterile technique, right? But normally with contamination, you're going to see something grow that wasn't supposed to grow. Contamination can also be considered where you're like, oh, I forgot to change my pipette tip, so now there's plasmid in the mix, where there shouldn't have been plasmid. And so there are things, you know, some of the things you're doing your best to control human error. It's going to happen. So that's why you want to be able to repeat what you've done. And the only way you can really repeat what you've done is by writing things down. Because you want to do it the same way. Because it's really unfortunate when you see something and you're like, great, and then you can't do it again. All right. So like I said, it gets a little more complicated as we go further down, but hopefully that's going to be on the video. So, okay.
So the amp resistance is on the same level during the active.
Hold on. I'm going to ask you to ask that one more time. Okay.
So the amp resistance is on the same level that you're trying to do during the smell.
Yeah, exactly.
So it's one level.
Yeah.
So is there any way to go back and give it ampicillin non-resistance?
Yeah, you can interrupt the ampicillin gene.
Hey, Mary, can you actually repeat all the questions? Because the microphone's not going to pick it up all the way back there.
Yeah. The question was if the lemon gene is on the ampicillin-resistant plasmid. The answer was yes. And the second question was, could you remove the ampicillin gene? Yes, you could. And there are 2 ways you could do it. One, just simply cutting it out with some enzymes. You could also interrupt it like we did the LAC-Z. You could also just, now that you know your gene works, you could put a different plasmid. You could also use PCR or some other method. PCR would work like this. You would have your whole gene in here. Your ORI, your promoter, and then your cool gene of interest. And then you have your ampicillin resistance. So, PCR, you design primers, and based on the directionality of the primers, let's say if you just want to clone out this gene and make some primers, this, and like this for the top and bottom strand, and I would only get this piece out. If you're going to think about flipping that around, and making it so the primers go out and away from the area that you don't want, you would only get this product and not this piece.
So you would wait to get a population for saying that has so many years history of working on target you're trying to make.
If they get so I was asking if I'm waiting to get a population that has both the ampicillin resistance and the gene of interest, right? Basically, I'm the only E. coli that's going to grow is going to have this combination. If they don't have this combination, so this gene is being put into this vector here with ampicillin resistance. Here is that plasmid. E. coli that get this plasmid will make antibiotic resistance. Those are the only ones that are going to be able to grow on the media. Now, once I remove ampicillin resistance, it's going to be really hard to choose which one. And so if we do want to remove ampicillin resistance, we're going to have to use some sort of differential selection. But part of the problem with that is it's a lot easier when a good part of the population dies, when they don't have what you want, because it's finding a needle in a haystack. Because when everybody grows, you have to put a much smaller concentration of much smaller concentration of E. coli on the plate, because otherwise it'll be a lawn. Everything grows. It will be a lawn. You will not be able to choose one single solitary colony. So you have to plate a lot more bacteria and look through a lot more. So instead of just plating maybe 3 plates, you will plate like close to 100 plates. You'll have to do the math in terms of the efficiency. And then you might not find it, but you keep the reaction and replate again. Yeah, Marie.
So I have a question from Jeff. He said, is a single blue-white screen sufficient, or should we plan on doing more than one? I believe that's in the experimental control.
You'll need a different, so the question is if you'll need to do more than one, let's say differential screen. The blue-white selection is a differential screen, so it doesn't kill the organisms you don't want. It just makes them look different. And so I'm not exactly sure to what end Jeff was referring to. We will only need one differential marker per gene. I would recommend having an additional differential marker if you're going to be adding more than one gene, because it's going to be hard to determine. If we were moving along to adding our second construct to have the full 2 genes that we need in there to make a lemon smell, we'd only have to have one difference in this process, and that would be choosing a different resistance marker. So we could use kanamycin instead. And so we can still use blue-white selection, all the steps to be the same, until we did our next step. So we would be doing a transformation, and then this E. coli would already be in there. So our transformation would have an E. coli that's been transformed.
Some information may be on their website as well, and then you can have
That's going to have our one of our genes of interest, right? So we're going to start off with the one that works separately or whatever. And that's ampicillin resistance. And then we're going to get into our second gene. It's fine. Be creative. That has our second gene of interest, which is represented by this brown, which I'm going to make a box. And then kanamycin resistance, which I'm going to make that a box too to differentiate it. So this E. coli, we would know it had both the plasmids because it's plated on ampicillin and kanamycin.
You need somebody who can take the lead, not necessarily, right?
Plates. So you can, the moral of that story is you can put 2 different antibiotics on a plate. Some plasmids will have 2 different antibiotic markers. I hope that answers Jeff's question. If not, they'll need to restate it kind of differently so I can understand it better. All right, so cool. So just because we saw colonies grow, it doesn't mean that we actually got what we got, right? So the first step, so yay, colonies grew on AMP plus, and all of our other false positives, fine, it's cleared out. When I use the word colonies, can somebody remind us what that means? All right.
Bunch of bacteria.
Yeah, so colonies, yep.
Separate groups of bacteria.
Yeah. So a colony is a group of bacteria from one, let's call it a mother cell. So that means all the colonies grew from one cell originally. So there should be clones. Okay. So that's what I mean by the word colonies. Okay, we got colonies. Yeah. So one of the first things we're going to do is we're going to grow these cells up and then do a mini prep. So sure, they grew, right? We're going to make sure they actually have DNA, like a plasmid-like DNA in them, right? About the right size. Just see. So first thing we're going to do a mini prep, and then to see that the mini prep got us something, we're going to do a gel electrophoresis. This is going to make sure that the plasmid we're seeing, how about put it in there, plasmid is there. It's also the right size. Okay. A couple
What do you mean
options.
by the right size?
Well, so when I say size, I'm talking about the number of nucleotides present. So all E. coli have DNA in them, but pseudomonas DNA is much larger, so you're going to expect to see some DNA. And so that's why I'm saying the right size. So you'll have a DNA ladder, and that ladder has pieces of DNA in it of different but known sizes, and you can kind of compare. We'll be doing a gel electrophoresis again on Sunday, so if you want to see another example of that, we'll totally be doing that together. Okay. The second thing that we might want to do is PCR. And we'll do probably PCR of either our AMP gene, right? Or anything, any part of this that might distinguish this plasmid. It might be convenient to make primers that go here and here around where we might insert our gene of interest, right? So that we'll have those ready for later down the pipeline, and we want to make sure that the gene of interest also got in there because we do this exact same process, right? So PCR, this identifies our plasma. So just because we got a band on our gel doesn't mean that it's still the right plasma. We can double check with PCR, do another gel electrophoresis, and the band that we see should be the predicted size between our two primers. But let's not even finish there, folks. We can also send our plasmid or our PCR product for sequencing. So this is more important when we're actually putting a gene in that we're interested in. But if you see colonies, right, that are growing ampicillin and maybe like are white, right, you're definitely wanting to do sequencing. So there's additional steps before and in between, doing your macro steps. When I say your macro steps, those are the plates that you're putting bacteria on and seeing if things grow or not. So if you can imagine it can happen, you should probably make a test to prove that that's not what's happening. Because science is about the process of imagining, disproving your idea somehow in the physical world. If you can't possibly imagine a way to disprove an idea in the physical world, then it's not a question that can be answered by science. Yeah. Are there more questions?
Just a basic one. Is laxity innately in E. coli, or is that a special order item in bacteria?
So, E. coli can break down lactose, and I think they do have that gene set in there. I'm pretty sure, so here that I'm qualifying, I'm pretty sure that the beta-galactosidase has either been changed, so that's the gene that Laxi codes for, beta-galactosidase. It's either been changed, so it reacts with XGAL, the substrate that turns the E. coli blue, or XGAL is just a special thing that we've found as an indicator that works on the sheet. Now,
So it's
the competent cells that you should be using this in should not have LAC-C. It should not have that gene to innately make blue. So not naturally make blue when plated on XGAL. You want to make sure that that's not happening. But I can't clearly, I have to look to see what the difference is. I'm not sure. Yeah.
So if you set it up for sequencing, do you sequence the whole thing, or do you tell them I'm looking for this specific?
Yeah, I just don't want to stack these.
So the question is, should we, when we go send stuff off for sequencing, do we just tell them sequence part of it? How do they know what to sequence? For a plasmid, you can certainly give them the two promoters that you have, right? But generally, I guess for a plasmid, you can just use their in-house promoters. There's some pretty promiscuous promoters out there that will work, or primers, sorry, promiscuous primers that'll work almost on any sequence. And then they'll pump you back a sequence and you can compare how it looks to what you'd expect. And then, but generally what I would do is I'd just be looking at our PCR product. I'm pretty sure if there's something else really wrong, I'd look at my plasmid when I get it sequenced. But your PCR product will be delimited by your two primers. And so it'll sequence this way and this way. So I told you about, sorry, I was pointing up here when I was talking about sequencing your whole plasmid. Forgive me. They would be oriented a different way for your, I don't have arrows on these, so they'd be oriented a different way. They'd be pointing inward here for your gene and outward for the whole plasmid, or they'd be inside the gene. I would only sequence my PCR product, pretty much, because that's the thing I'm most interested in. It's enough to identify that, yes, indeed, this is the plasmid I thought it was. And it's also where I put my gene of interest, so I can just say, yes, my gene of interest looks like it's supposed to. The sequence is in here that I thought wasn't there. Because that could be a reason why your gene is not being made. Yeah?
So, like, antibiotics, like penicillin and kanamycin, they have a shelf life, and what effects can have to be kept at a certain temperature? Is there any quick tests to tell if they're good or not?
Yeah. So the question is, how do you tell whether your antibiotics are good or not? The only way you can test is if they kill things. And so you keep them stored as it's recommended by the distributor, right? You can look that up online to see what the good shelf life is. Some can be kept at room temperature, some can need to be kept cold. Normally, when I've already mixed, so made a solution because antibiotics tend to come in a powder, and so you need to mix that powder into water, methanol, ethanol, whatever it dissolves into. And then once I've done that, I keep it in the freezer. To test to make sure that the antibiotics are working, you're going to grow cells that don't have a resistance to that particular antibiotic on a plate with the assumed appropriate amount of antibiotic in those cells. It's also nice to know that it's, just because those aren't growing, it just could be kind of enough to make it so that cells without ampicillin resistance don't really have it, or it's harder for them to grow, or it's just slower. It's really nice having a positive control. You're like, these cells should, or I guess you actually a false negative, sorry, right? That these cells should be growing on this ampicillin dope plate. More questions?
Let's ask you from a practical standpoint, if we were doing this experiment from scratch, could you just start with a plasmid that has no AMP-R cassette, and then just say my gene of interest is an AMP-R cassette, and then append it to that a gene of interest? Do you know what I mean? And this way, like, and then just select for that, and not have to worry about GFP or anything.
Yeah. So, but the problem with doing multiple steps at the same time. So yeah, absolutely.
Right.
Is you're like, okay, it didn't grow. Why didn't it grow?
That's when you begin crying.
And you're like, okay, I don't know. Then you go like, cool, well, I'll do all my controls this time.
Yeah. Back to it.
Or I'll do it stepwise. So and there's so many things that could happen. That's why it's preferred to just, even though it takes longer, you know, right? You know what's going on, and you can start troubleshooting with the actual problem in the very beginning, or asking for advice in the very beginning, rather than being like, cool, I think everything's working, and then it's actually nothing. Or you're trying everything and nothing's working and you don't know why.
Have you seen or used those automated kind of boxes that do a lot of this stuff? Like you'll essentially tell it in out, petri dish here, put it on a conveyor belt.
Yeah, there's a lot of labs on the cloud that cost money. If you're doing this as a hobby, your time is free.
Sure. But those exist, I'm guessing, for like more institutionalized. Like if you're doing this as a business kind of thing.
You want, the thing is though, for one of the companies I work with, we used to use a service company for building constructs. So when I say constructs, I'm talking about vector plus gene of interest, right? And build a variety of them. But you still had to troubleshoot. Sometimes it wouldn't work at all. Sometimes controls wouldn't work and you didn't know why. Right? And so you're like, what changed? And so you buy all new reagents, and still has some of those human aspects in there. But it is nice for reproducibility to be able to utilize the robot. So because you're using, like, say, if you're doing 96 reactions, and then you have that thought, and you're like, wait, was I at 81 or 82? Normally what you do is, if you have an idea, you're going to skip, right? Write down in your notebook, and then just carry on. You're like, well, I'm not sure. I can just retry, just make a note that that's where you made a mistake, right? Or if you're like, I don't even know where I'm on that row, skip that row, and then start over. You know what I mean? So there are ways. You just make notes of it in your notebook, right? Because you could find something really cool by a mistake, but you can only know it's cool if you write down what happened.
I remember that, like, when we were doing the labs and you asked if anybody had thought that they made a small mistake so that we could write it down, and if it worked anyway, we have a discovery.
Yeah, or be like, know what's like allowable in the human error. For instance, when we were doing the mini prep class, when we were extracting plasmid, I forgot to tell you guys for the very first added stuff that you need to resuspend the cells. Now, that could have affected our yield. That could have yield said how much plasma we got. We still got some, but not as much. It could have also affected whether we got plasmid at all. It also could have affected the quality of plasma that we got. We could have harmed the process somehow. And so the first check is like, how much plasma did we get? Well, everybody got plasma. So we, at least I'm thinking that at least a third of the class weren't, you know, I either had no experience or weren't just like mixing things up because it sounded like fun even though I didn't tell you to, right? Hey, that's still okay. Everybody got plasma. There were different amounts of plasma in each yield, but there's issues with the pipettes as well. So there's some things going on there. To understand the quality of the plasmid, really what we need to do is a transformation to see if indeed the plasma that we harvest had the ability to give new abilities to just a good old confidence cell. But we could have known that, yeah, it doesn't really matter if you don't suspend that pellet of cells. It doesn't matter. Yeah, you will still get plasma, but you get less, or it just doesn't matter. In fact, it really matters even which order, or like you could switch things up. You always try it, like, oh, I messed up, right? How you messed up, and you'll see if it actually matters, or what effect it does have. Are you guys ready to talk about overarching concepts, or do we have more questions? We have more questions. It's okay. All right. Overarching concepts. Use the funnel method. So first off, can you science it? So that's the question. Basically, is your question valid? To answer this question, this is just all imagination. You need to think of one way. In which your idea can be proven, sorry if that's yours, wrong in the physical world. Okay, so can you science it? What kind of questions can we actually ask? So there's tons of questions out there, right? Can you science it? Is there a way for us to prove in the physical world, see, that gravity keeps my feet on the ground? Is there a way to maybe falsify that? That's probably a hard one, right? Is there a way it could be falsified? We just have to imagine, right? Let's say, I could be staying on the ground because I have tape on my feet, right? That'd be one way that I'm sticking to the ground, literally, right? So that would be a falsified way that's not gravity, that's actually tape that I'm using, right? So we can make a test, okay? Because that's for a lot of things, right? Some things are more practical than others. It's not that everybody has tape on their feet, for instance. It might be something else. That would be one way that I could falsify, right? Or think of another explanation as to why. But there are plenty of other questions out there that are really a lot harder to imagine a way that you could falsify it. Yeah.
I like to add on to that. You keep saying about the negative approach to science.
Yeah.
But it's important to remember that it's, no matter how much negative things you do, you're not saying it's true because the wrong thing went wrong. You're reducing the uncertainty of the thing being wrong. Like you're more and more certain that you're not proving it's right. You're more certain that it's not wrong.
You're reducing the uncertainty, right? So you're saying, well,
Sorry, but can you repeat the question?
So no, it's actually more just a comment. The comment is that let's not think about science as being so negative. I mean, come on, good vibes, folks. What we really want to think about when we're going through this process of negatives is we're actually putting the question to an easier scope. I mean, right now, when we first have a question, it could be anything. It could be anything explaining it. And when we start seeing what's not explaining what's going on, we have a better idea. We're actually more certain. So we're actually headed towards certainty, headed towards a certain level of truth, right? And so that's why we keep on going like, I'm proving this part wrong, I'm proving this part wrong, or I'm proving that this idea is not valid. But what really science says is that we're trying to show that each one of these things that we're seeing is not just due to random chance. Because, man, anything can happen. There's a random chance, anything. And so instead of it being random chance, we want to know that it's not random chance. And that's where we're trying to get more and more certain. So next thing is, all right, cool. I can think of at least one thing. Move on to what do I want to see? And I'm saying see rather than prove because one, I want you to remind you that you're going to have to look for something physical, right, that we're looking for. It's going to have to be some sort of test. Also, it's impossible to prove anything. You can just be more certain because there can always be something else that's going on, some question that you haven't thought about asking. Because it's hard to even think about questions sometimes. In fact, when you disprove your idea, it just leads to more questions. And so, what do you want to see? So, for instance, like I wanted to see, that my plasmid got into my E. coli, right? And so, and what I actually see is colonies on my plate, right? And so, I wanted to think about all the reasons why I might see colonies on my plate. What are all the reasons that I could see colonies on my plate? What I don't want to see. So this is all of the stuff that is like not seeing colonies, right? You want to think about explanations as to why you may not be seeing what you want, right? So those controls can be in there as well. And then you build your experiment out. That's the overarching. Can you science it? articulating what you want to see, articulating what you don't want to see, and the reasons why those things might be true. And that's experimental design. That's it. You're it.
Our last lecture is over bioethics. Now that you've created this dangerous class of
Bioethics?
super
No,
guns.
we don't have another lecture. No, yeah. But then we have, we just, this weekend we have another lab where we actually use restriction enzymes and we see what it looks like on a gel. It's like doing a little bit of forensics. So.
I was telling my friends I was making genetically modified glow-in-the-dark E. coli, and
Well, yeah, you are. And what'd they say?
They're the hit of the party, man.
yeah, people were like, "Wait, what? E. coli? Isn't that dangerous?" I'm like, "No, it can't make you sick. It's like a little poodle dog E. coli."
So what you meant to say
Yeah,
was,
it's really
I
cute.
know, right?
I heard to pass this around a lot, but don't just pass around the lecture, but also
Well, no, there's a lot of startups that are out there doing it. There's Synthesia and stuff.
Oh,
Yeah.
yeah.
Oh, well, I don't know how common it is, but I know it is. Or it's being They're easy to go. They've already done it.
No.
There is no attendance sheet.
Oh, okay.
I want to say, what is it? Is it like for somebody to be assigned to big law, it has to be like 6 Sigma certainty or something like that?
No, you just have to get enough people.
Six Sigma is more of a production. Like philosophy. I kind of run out of material for that. I think that's the best.
Can you get to 99.9999% certainty in production? I've never heard of anything
So, for
that's
example,
certain.
if you like design something.
And like, I can think of math equations that are less certain.
If you look like and you want to and you look at all the components in that stuff, right? Then there's a probability of start searching for each component in itself to get you to that.
Oh, I guess that makes because you multiply probability.
So that's where the Six Sigma comes out of.
That's astronomically high though.
Exactly right. So when each part has such a huge percentage, again, applying all that probabilities.
Yeah, so my first proof of concept.
What's the general accuracy of like technical process? What's the sigma probability that if I take the instruction, return the wrong result?
Well, you have alpha particle upset. Right.
Right. What's the probability factor?
You're an
I want to say it's something like if you have
on satellite, it's a big deal.
Well, it's like satellite, it's a big deal. It's like it gives.
And they have her.
But like if you have 16 gigs of memory, I want to say like 10 bits are flipped by cosmic radiation.
Oh, like at the base maturity. I worked on this problem for a satellite. Like, where is it?
Oh, well, I know. So
Air detection correction. What is down here? No, that's a problem.
But that might be a good idea.
Absolutely.
Sorry. Yeah, I know that you're working
The lecture video from last week was super helpful.
It's on Marine's account, so just ask. Oh, okay. I'll ask. See if you can like be able to get it to me.
And also right now is I'm not really sure what's going on.
Try to organize it. Oh, okay. But if you have questions, you can always direct them my way.
Okay, cool.
I'm happy to answer shit.
Great. Alright. I wanted to do the desalination workshop. I I don't know if that's public.
My project, yeah, I'm going to be restarting that.
Oh, okay, cool.
So you can join me and help me out, and I can just like.
Okay. Okay, cool. Yeah, I'll be showing up and stuff. So I start my second semester at OCHEM in like a week. So my hopefully should stay high. And
Excuse me.
Probably a little lower, but yeah.
Does all of this mean that,
Oh, is that gonna be on the meetup then?
Yeah, I might, but I'm just gonna like schedule on my own time.
Okay.
And just like, so it's not, I don't think it's gonna be super consistent, so if I have people that I know want to come on, it's probably like, say, hey.
Okay,
Yeah.
cool.
It's all gonna be open source and everything like that. It's just sometimes it's hard to do teaching and also getting shit done.
Yeah, oh yeah, I believe it.
So that's one. Show you how to do stuff, and then because my idea is to be able to help train people so they can kind of work either autonomously or pick where Slack off or just can work in parallel, so next to each other, so they get more stuff done.
Cool. Is the installation somewhere? Is that lab or is that a lot of big research?
Well, I've done a lot of background research, and I'm happy to share what I've brought up. I'm going to be talking to one of my friends who does a lot of design and creative stuff. So we can just kind of play around with
Okay.
because I want to do a directed evolution first, which to make it more
I don't think that's what's grabbing
yeah.
it though.
And then playing around with just putting in a bit of like, well, that's the long term, getting that off the ground and working. So this is the first part, and then
So you're trying to get bacteria to desalinate water?
that's right. So first we make sure that I'm gonna make the Stenococcus that I've chosen already some pretty salty media, but want to feel because we're asking it to take in all the salt from the outside and not that. All right.